So where are we, doctor? We are talking about the caspices. So we are talking about intrinsic pathway, right? The bottom line is backs and back are basically pro-apoptotic is what we are talking about. Now what happens in intrinsic pathway? It is the mitochondria which is very important to mediate the intrinsic pathway, from which the cytochrome C become released. As we have seen, it lead to formation of an apoptosome complex, which ultimately lead to the caspases. Ultimate executioners are basically caspases. How it happens? There are two different pathways. So in the case of the intrinsic pathway, the cytochrome C get released and it will bind with the Apoptosis. After binding, it typically will create the pro-caspase 9 to get activated, and that activated caspase 9 will carry forward the caspase cascade is what need to be basically remembered. So where do we get Apoptosis, doctor? Basically, is it pro-apoptotic or anti-apoptotic? Pro-apoptotic in the intrinsic pathway. That's what we need to appreciate. Then there are some other mechanisms by which apoptosis is mediated. There is a cytotoxic T cells are there, no? They become activated and they lead to the activation ultimately of the caspases through a mediator called as granzyme B. So what is granzyme B? It is one of the proteases released by the cytotoxic T cells is what you need to basically remember. Once this granzyme B enters into the target cell, then the entry of it into the target cell is mediated by a protein called perforin, which is also important to be remembered in the apoptosis. Now in the apoptosis, what happens to the DNA? The cell shuts out down, but DNA is the most important thing inside a cell. It is not an easy thing available. Number one, it decides the intelligence of the future generations, right? So what is the fate of the DNA in apoptotic cell? There is a very important characteristic appearance of the DNA called laddering, like a ladder. Laddering appearance of the DNA. Are you able to see a ladder here? Typically, whenever you do electrophoresis of the DNA, this laddering appearance of the DNA on the electrophoresis is a very, very characteristic feature, is what you have to ultimately remember. So, why this laddering occur? The DNA become degraded by the endonucleases, ultimately into the nucleosomal chromatin fragments, and 180 to 200 base pairs of multiple fragments will form, and whenever you do electrophoresis of the DNA, laddering pattern of the chromosome is a very classical feature. But do you see it only in apoptosis? No, sir. Many other scenarios you find it, but it is characteristic of apoptosis, not specific for the apoptosis, is what you need to remember. Then transglutaminases, they also have a very important role in apoptosis. What is that? Basically, inside the cytoplasm, the various proteins in that apoptosing cell, there is a cross-linking done between them, which is responsible for the shrinkage of the cell, is mediated by the transglutaminases in the pathophysiology, is what need to be remembered. So doctor, what is the way to know whether a particular cell is undergoing apoptosis? Is there any way to prove it? Tunnel assay, or otherwise you may say tunnel assay, because A for apple, B for boy, C for cat, in that book these phonetics are not defined. We can call it whatever based on our vernacular slang. Tunnel or tunnel assay is a way. What is the meaning of it? The terminal transferase DUTP nick end labeling, it is called as, is called tunnel assay. What is the use of this? You can basically quantitate the apoptosis using tunnel assay, tunnel or tunnel, whatever you call. So using this, you can know how many nucleosomes after cleavage of the main DNA have formed. Based on that, you can be able to quantitate the amount of apoptosis. Similarly, caspase assays are available. More caspases are there means there is more amount of apoptosis. That's the meaning of that, as all of you know very well. Is it echoing? We have too many inborn errors of metabolism. I hope echo, can you slightly decrease that speaker? Slightly. Yeah, that may bring down the echo. Please punch, is the echo cleared? Anita has got a special echo heard. Is it heard for everybody? Then we have universal echoing or only specific echoing. Yeah, that's what, audio only will echo. Video also, if it is echoing, then we are having LSD and hallucinations. Huh? Prashant says no, no for echo or yes for echo. Clear, very good. I think Anita need to set her audio little well. It is only a focal echoing happening into the Anita's ears, not into Prashant or anybody. That's right. Harika also says clear, very good. So doctor, what genes regulate apoptosis? BCL2, BAX, BCL2 stimulate apoptosis. Then which gene product inhibits apoptosis? BAX, BCL2 inhibits apoptosis, BAX facilitates apoptosis, P53, what will it do? P53 product will decrease the transcription of BCL2 and increase the transcription of BAX. That's the reason P53 facilitates apoptosis. That's the reason any P53 loss like Life-Romanes syndrome, there is nobody to facilitate apoptosis, then apoptosis doesn't occur, then tumor will grow. That's the reason there is a tumorogenesis in P53 loss in Life-Romanes syndrome is what need to be remembered. Now doctor, necrosis versus apoptosis, what is the difference in their in the biochemical changes which we have discussed? Necrosis is a passive form of cell death. It does not need any gene involvement or executioning or regulating genes. मरने के लिए परमिशन का जरूरत नहीं है. So necrosis doesn't need anything. Then the DNA fragmentation is basically very haphazard rather than very regular ladder pattern. You don't see. What do you see in DNA, which is in a necrosing cell? If you do electrophoresis, smudge pattern is what you typically come across. Marked amount of inflammatory reaction is a thing which you see in necrosis, is what need to be remembered. Are still doubling audio? Is the audio clear with others? Yeah, just ask someone to help out with the audio to Anita, because others are not having a problem with the audio. Alright, some days are bad days, tough days, no bad day. We are alive means it's a good day. So, tough days. Now doctor, marked inflammation is necrosis. Lysosomal enzymes eating the cell is a process seen in necrosis, not in apoptosis. Right doctor? Then disruption of cells, involvement of chemotactic factors, involvement of phagocytic macrophages. Macrophages are also involved in capturing the apoptotic bodies, but more they are involved in the necrosis. So death by injury is necrosis, death by suicide. My life is enough. Cell will decide, that becomes apoptosis. So the calcium influx, cathepsins, caspases are also the part of necrosis, but all the inflammatory mediators, they are all the things which are seen in the necrosis. So doctor, ultimately in the literature given to you, I have given you the differences between apoptosis, necrosis. Ultimately examiner will ask point to point each of them. You need to have a clear-cut mastery. I don't want to go through this table because it's a repetition of what we have done. So that's all the story, doctor, about apoptosis.